Hello, everyone. Welcome to our next installment in this LetPub webinar series. Today, as you can see here, we have illustration is key, preparing eye-catching figures for publication. My name is Dr. Clark Holdsworth. I'm the Senior Manager, Communications and Partnerships over at LetPub. You can see here, I'm joined by Dr. Avriel Licciardi, also of LetPub. We're going to introduce ourselves in brief here coming up. I know a lot of you are probably new to this, so before we do get started into the content, I'll be sure to cover a few housekeeping items, common questions that you may have, how these webinars run. But first, we'd like to introduce ourselves for the people that are new. I know a lot of you are joining us back. We do appreciate you returning to the series, so hopefully those of you who are coming back are finding enjoyment and a lot of value in this series. We've got quite a lot of participants today. We're nearing about 500, so I'm going to be sure to go over how the Q&A works because it looks like we're going to have several questions. As I indicated, my name is Dr. Clark Holdsworth, Senior Manager, Communications and Partnerships at LetPub. I do have an academic background, as does AV, which she'll outline for you. So we're both coming from academia. I have received my doctorate in physiology from Kansas State University. My focus was mostly on preclinical models, cardiovascular mechanisms, particularly of dysfunction, like chronic congestive heart failure. So a lot of focus on the oxygen transport chain, all the way from the ambient air down to the very low partial pressures of oxygen and muscle myoglobin. So lots of preclinical models, as you can see, very life science oriented. That comes into play in a lot of the content that we present here. You'll see that on AV's side when she introduces her background. Ours juxtapose as well, but you can see in our responses in the Q&A, that type of thing, where we're coming from. So we hope we're able to cover based on most of our backgrounds, the various things you'll see. A lot of my focus is on statistics, study design, definitely scientific writing, obviously, is a big component. So this is my big focus from my previous background. So AV, I'll let you introduce yours.
All right, well, thank you, Clark, and hello to everyone. And again, thank you for joining us, whether you've been to part of this webinar series before, or perhaps this is your first time checking in. Again, my name is Dr. Avriel Licciardi. You guys can call me AV for short. I'm the Research Communications Strategist here at LetPub, and I'm also a Senior Associate Editor. As Clark alluded to, my background is also from academia, however, quite different academically speaking from Clark's. I'm a geologist at heart. I focused on reconstructing glacier changes throughout the past about 25,000 years, both in Chile and Peru during my master's, and then up in Greenland for my PhD. Since then, I jumped ship to scholarly publishing, and I focused a lot here on outreach, specifically for international authors. As Clark said, we're both very excited to bring our diverse backgrounds to you that cover both life sciences and physical sciences, and how that plays an important role, giving you insight, at least in terms of how to create these eye-catching, visually appealing figures that are so critical to getting published, which is, of course, one of the most important goals of being an academic and a researcher in general. So I'm going to let Clark go over some housekeeping for us before we get started. So Clark, I'll hand it over to you.
Yeah, the most important part of my job for these webinars, obviously, we have a few common questions that we get, so I want to cover those quickly. First of all, how the webinar works. We're probably going to run about an hour is what we aim for, and we'll have a robust Q&A session, which I think is definitely the highlight of these webinars. During the webinar, as many of you have already found, there's the chat function. So you do have the chat there. Obviously, we have people who are occasionally running into trouble with audio and everything. It seems like we're running okay today. So just a reminder, if you are having trouble, usually you can just go ahead and refresh the page and you shouldn't have any issues. Always make sure that your audio is on your computer, but it looks like on our end, it's working quite well today. The other key feature that I want to point you to is the Q&A. So there's a Q&A button down there. You can leave your questions here. I will be looking at the chat, but there's a very good chance that we won't see your question in the chat, and we'll only be answering ones from this Q&A function. So go ahead and enter your questions there, and feel free to start now. You probably came into this with some questions. You can go ahead and enter them. Within the chat, there's the ability to upvote any question. Please utilize that. Today, right now, we're just under 600 participants, which means we're going to have a lot of questions, and we're going to have to curate it a little bit. So what I want you to do is make sure you upvote questions that are similar to yours, or that you think are important, and that helps us to curate it, go down the list for the Q&A session. As you can see through PeerRef, we also do have a hub for this. So we do want to answer all your questions. And what we're able to do is after the webinar, if we haven't gotten to anything, or if you have follow-up questions in the future, or even if you just want to jump in a discussion with us, you can go to that PeerRef hub, where we'll be able to answer questions with you on that discussion board all throughout the week, all throughout the coming month. You go ahead and leave things there. We're checking it all the time, and we can get to your questions there. So please do join that afterwards. I'm going to go ahead and once AV starts, leave that link for it in the chat so that you guys can have it. I'll do it again later on as well. The other items I want to cover are webinar certificate. We will provide that. So what's going to happen is there's going to be a follow-up email. You will get access to a webinar certificate that's in PDF form. We can't alter that for anything specific that you may need, but if there's any questions about it, you can go ahead and send those to us via email, but you will get that in a follow-up email. Usually it comes out within about 24 hours. Also in that email is going to be this full recording. All right, so not the slides for the PowerPoint, but this entire recording will be available to you on demand. So you're going to get a link for that, again, in that same follow-up email. So go ahead and just wait about 24 hours, see if you get the follow-up email. It should have both of those items for you. After that, if you have any questions or concerns, go ahead and reach out to us and we'll get it taken care of. So I think that's all, AV, for my housekeeping items, so I can let you get started. I'm going to be following along. I'll make sure we get the Q&A lined up. Yeah, and I think that's it.
All right, sounds great. Thank you, Clark. All right, so to jump into the content today, what you're seeing on the slide here is the outline for the material that we're going to cover. So if you've joined us before, you know that I like to start off with some intro and basics on the topic that we're discussing, which today, again, is crafting beautiful scientific figures that are both accurate and clear for the reader, reviewer, and editor. Importantly, we want to answer the questions of why are figures so important in publishing, and why we should be investing so much time and effort into creating them. Importantly, I want to review which parts of figures can be confusing from the perspective of the reviewer, editor, and reader, and of course, how you can go about avoiding having those sort of frustrations, which of course can lead to rejection of your manuscript. From there, I'm going to give you some step-by-step guidelines for figure creation. I've partitioned this out into 3 different kind of overall steps, from planning to prepping to actually creating these figures for your submitted manuscripts. And importantly, I also want to address how you can write effective figure legends. Again, figures are something that should be understood separate from the main text. So again, you can't just have a figure by itself with no legend or caption, as that's really an essential component to the figure as a whole. Lastly, I'm going to end with 4 kind of primary takeaways from this talk, as well as this extensive Q&A session that Clark alluded to. So again, if you have any questions related to the content of this webinar, anything related to scientific figure making, please input those into the Q&A function. And importantly, if you see a question from a fellow attendee that you would really like answered, you have the same question yourself, all you need to do is simply upvote that question in the Q&A, and the questions that have the most votes from all attendees will be the ones that Clark and I addressed first. So again, you do not need to rewrite the same question, just upvote a question if you'd like to have that answered. So let's jump right into everything. So the questions that I put forward for this webinar are, what makes a great scientific figure, but also what makes a bad one, and how can that be avoided? And as I said before, we want to answer the questions of why are figures so important for scholarly publishing? And the reasons for that are the ones that I have listed here. First and foremost, figures save readers, reviewers, and editors time and energy in understanding the core concepts of your science. They aid in understanding of your article and your research. And from a journal editor's perspective, and also a reader's, is that they help in reducing word count. So they make your manuscripts more concise. They might allow you to publish in a journal that has limits on word count that you otherwise wouldn't be able to get within those limitations. Importantly, many readers often only view the figures and the abstract, as well as the conclusion sometimes, before they decide whether or not your paper is worthy of their time for reading. And this is incredibly important when you think about submitting your manuscript to a journal for publication. Journal editors are going to do the same thing. They might also read your cover letter that you submit, but they're really going to jump right to the figures and right to the abstracts. So given that the figures are something that a journal editor is going to look at first and foremost, you want them to be as effective and clear as possible. It's also important to understand that if your figures are unappealing, unclear, low resolution, or low quality, that might kind of add a bad view of your manuscript right from the start. And this could be from a reader, reviewer, or editor's perspective. And unfortunately, if your figures are not well made and they're not well presented, it can tarnish an opinion of the work, even if the science is sound, which is quite unfortunate. So that's something that we definitely want to avoid. And as a primary objective of the webinar today is to teach you how to avoid that from happening. In general, all figures should convey information from the data that you're presenting both quickly and effectively. Again, a reader should be able to review the figure, maybe spend a minute or so looking it over and be able to start understanding the core message of that figure and its primary purpose in the paper. And while figures and manuscripts are incredibly important for publishing and for academia in general, it's also important for all of us to understand that graphics themselves are becoming more common than ever in academic publishing and the scholarly publishing industry. So again, not only are figures important for manuscripts, but you can be using them for graphical abstracts, which are becoming more common in publishing, perhaps your pictorial representations of schemes. So for these technical concepts that are made more pared down for the lay audience. There's table of content thumbnails that are becoming more prominent on journal websites, infographics, and then of course, you're able to use any of the figures that you create towards your poster presentations, your verbal talks that you present, or throwing them into sort of animations for any sort of video components that you're putting out, say on social media or using within your department. So again, collectively, figures really assist and provide that first impression of your article, that first impression of your research, and it's really important for you to focus on making that a good first impression. So the way that I've structured this talk is let's talk about the things that we want to avoid as researchers when we're creating our figures. So what makes a scientific figure confusing from the reader's perspective, from the editor's perspective, and from the reviewer's perspective? So again, these are what's going to cause people to not understand your figures. And these are things that we want to avoid. First and foremost, if there are errors in your data that can be easily seen by someone that's reading or reviewing your figure. This often leads to misleading representation. And while this may not be your intention, if that comes across to a journal editor or a reviewer in particular, that's going to kind of give them a bad feeling and they may reject your paper as a result. So again, first and foremost, you want to avoid any sort of errors in your data or misleading representation. Along similar lines, if there's missing or even incorrect information, that should be avoided, and as it can be quite confusing. Very common mistakes like this might seem trivial, but they're quite critical to make sure that you have before submission. For example, you want to make sure that you have axes titles on all of your graphs. You want to make sure that if a value has a unit, that's clearly represented. And if you're doing something that has images or perhaps geographical, you need to make sure that you have a scale. And these are all small little things that can be afterthoughts, especially when we're all scrambling to put things together. But they are part of those things that make your figures polished and put together. And that's something that's really appreciated by reviewers, readers, and editors. If you have poor color choices or unnecessary visual elements, that can lead to clutter and kind of take away the impact from your main data and message, which can lead to confusion. If there's any sort of messiness to your figures, that can also aid in confusion. This includes overlapping text, if there's erroneous lines or overlapping images. A big one for journal editors is if the font size that you use on your figures is too small, it's inconsistent in size or in type. Again, this kind of leads to that unpolished look or a low quality feeling that comes from the perspective of others. So that's something that you really want to avoid. Along similar lines, inadequate formatting is a big part of figure making, and we'll touch on this a little bit later. But again, you want to make sure that you avoid having any blurry or low resolution images. And also going to be touched upon later in this webinar is the absence of an appropriate and helpful figure legend. Again, you always want to have a caption, an explanation, or a legend explaining what your figure is saying to the reader. Again, this is going to really help in avoiding any sort of confusion. So collectively, these are kind of the main points that make a scientific figure confusing or unclear. And again, these are things that we want to avoid when we're making our figures. And with that, if any of these things are present, there's going to cause concern. And I'm going to kind of go through different types of concerns that may be felt by journal editors, reviewers, readers, as well as authors. And again, these are what these folks are going to be feeling when they're looking at figures that are unclear. So one of the primary journal editor concerns are improper image manipulation. And what I mean by this is sometimes journal editors run into issues where it's clear that an author has manipulated their data or falsified it in an unethical manner. So it's very common that once you submit your manuscript, you are asked by the reviewer or the editor to revise your figures to make them more clear. Authors often do this, and of course you do have to make adjustments and things like that to your figures to create them to polish them up. But there are sometimes instances where authors alter their figures in ways that are either inconsistent with their original version of their manuscript or inconsistent with what's required by the journal guidelines. And this can raise questions from the editors. And this is especially true if an author can never provide the original file for comparison. So the image that I've showed here is a pretty drastic representation where somebody added in a duplication in a new lane in their image here. So again, this is something that's very unethical and should be avoided. But again, if you use color adjustment and things like that to enhance something that shouldn't really be enhanced, that can also be a little funky. So again, you want to make your images polished, but you want to stay true to your primary data. One of the primary concerns from a reviewer's perspective is that a figure purpose is unclear. They don't understand why this figure was included in your manuscript. So this can be avoided by authors determining the purpose of all of their figures in their manuscript from the beginning. So for you as an author, if you are unsure of why you're including a certain figure in your manuscript, then it's very likely that a reviewer or an editor is going to feel the same way. So one of the most important aspects of this planning for your figures process, which I'll review in a few slides, is that every figure in a manuscript should have a clear-cut purpose. Again, space is limited in journals, and sometimes there are even figure limitations, so limitations on the number of figures that can be submitted. So you want to make sure that every figure you decide to put in your manuscript is really essential to telling your overall story. Again, what I have also listed here is that you want to address all of these issues before peer review, because many of us may be aware that responding to reviewers in your document is a time-consuming and tedious process. And if you address as many issues that you can foresee related to your figures before submission, that's going to save you a lot of time, especially if reviewers are asking you to recreate your old figures, create new figures, or revise figures where you might not have access to the original files. The last two concerns that we want to try to avoid when we're making figures is both a reader concern and an author concern. From a reader's perspective, if your figures are uninteresting, low quality, low resolution, or unclear, they might just think that the rest of your paper is going to be the same way. An uninteresting figure might be an uninteresting paper. So again, to kind of highlight this point that most readers of your manuscript are going to assess the figures and the abstract before reading your entire paper. So if your figures give a bad first impression, those readers are going to be less inclined to read past your abstract and the rest of your paper, which you've put so much time and effort into drafting. Again, this is so important. Figures are so important to draw the reader in and really have them take home your primary message of your research. And as I've kind of alluded to with all of these concerns, a concern for us as authors is, of course, wasted time. We don't want to waste time. We don't have time to waste anymore these days. So while it may seem like it's time consuming to create these effective, visually appealing figures from the beginning, it's incredibly important to invest that time, because you're going to be likely spending way more time revising your figures, recreating, or even correcting some of your data in order to publish later on. So again, putting the effort and time in at the beginning will likely save you a lot of time in the long run later on. So now that we've gone over all of the aspects that can make a figure confusing and some of the frustrations and concerns that those confusing elements can lead to among all parties involved in publishing, so that would be the journal editor, the reviewer, the reader, and yourself, the author, let's jump into what we should be doing to make our figures beautiful, accurate, clear, and visually appealing. What I like to think about is that all figures should be achieving certain objectives. And some of these are going to be scientifically, and some of them are going to be putting forward, again, just a visually appealing figure that's clear. So each figure in your manuscript should have a purpose, and it should achieve the following objectives. First and foremost, figures are going to be documenting your data. And if you have some sort of geographical component to your figures, they're also going to be documenting your study location. So again, the first and foremost, most important rule of a figure is displaying your data accurately, clearly, and concisely. Another objective for all your figures is that they should be telling a story, or perhaps a part of your overall story that your manuscript is putting forward. So you must identify the story, or in other words, the big picture message of your study before creating figures. And this is hard, and it's on an individual basis. But for me, my research advisor always had us create our figures before we even started writing our manuscript. And at first I found this really difficult, because all I wanted to do was get started with writing, and the figures would come later. But it is way, way easier to write if you create your figures first. Because that really does help you identify your story and how each of your figures will fit in, and how you're going to kind of arc your manuscript from start to finish. Again, by identifying what figures you're going to create first, and then creating them and then writing, this really ensures that every figure that you're putting in your manuscript does indeed have a well-defined purpose. And again, this is important because many publications limit the number of figures due to space constraints. Other figure objectives are that if your data has any sort of cause and effect relationship, you want your figure to allow the reader to make inferences about those relationships. So that means that you need to include information about both of the variables, which are your independent and dependent variables, in the figures for the reader. Again, making this clear and allowing the reader to come to their own conclusions. Along the same lines, you want to make comparisons in your figures, or allow your figures to have your data be able to be compared. For example, this would be displaying trends between variables through time. Importantly, well-presented data should encourage the consideration of not only your preferred opinion, but also alternative explanations. In other words, you want to try to allow your data to speak for itself, rather than kind of forcing your preferred opinion on the reader. So try to be as objective as possible. The last figure objective is that figures should be integrated with the text to enhance the overall communication of your paper. So figures should complement the main text, and the main text should complement the figures. But again, because people go and look at the figures first and foremost, they should be self-explanatory, and they should be understood independent of the text, which again is where those figure captions or figure legends can really come in and assist. So while these two counterparts, the main text and the figure, should be complementary, it's also really important to not repeat the contents of your figures within the main text. This is something that reviewers and journal editors are really going to be looking for. Again, they're trying to save space and trying to avoid redundancy. So again, what I recommend is that you use the text to focus on the significance, or the key points or key trends of your findings, and then the data is going to be more intricately displayed in your figures. So these are the objectives that every figure should be striving for and that you should be striving for when you're creating your figures for your manuscript. So the question that all of you may be having right now is, how can I achieve all of these objectives? It may be quite overwhelming. So what are the steps that I can do to move forward in that direction? Step one is planning your figures. So again, what story do you want to tell with your figures? What is this overall big picture message that you have with your manuscript? That take-home punch that you've been working so hard on for your PhD for the last 4 years, or the last 15 years if you're a faculty member. So planning your figures, I want everyone to take a step back and realize that this needs to start way before you've collected all of your data and you're sitting at your laptop ready to start writing your manuscript. This should start in the lab or in the field if that's a component of your research. So again, what aspects of the data will ultimately become figure material? If you're in the medical sciences, you're probably going to be taking a lot of photos on microscopes that you're ultimately going to be putting forward as images in your manuscript. For me, as a geologist, I would need to be taking samples of the geomorphology in my field area to illustrate a concept, or the samples of the rocks that I've collected. Now, a lot of this you may be thinking, oh, I can't retrospectively or retroactively go back and fix this. But again, learning this now is going to be important because you're likely going to be continuing on research in the future, doing new projects that you can implement this. So again, think about it as you're reading papers. You'll probably see that there are similarities in data that's always represented. Western blots are always going to be put forward as images and made into a figure in papers. For me, rock sample pictures are always going to be there. So because of my research I've done in the literature, I'm going to understand what figures that will be ultimately expected, and therefore I can start planning my figures much earlier in time, again, in the lab or in the field. The second comment here is to master your technique in planning your figures and creating them. So again, high quality images often require the appropriate equipment. Again, that might be a high quality camera that's attached to your microscope, a nice camera in general. Again, this is really important. Your pictures are your data. So when you're planning your figures, you want to make sure that right from the beginning, you're able to save your figures at a high resolution and in the right format. And this is really critical. I'm sure that many of us have spent hours searching on the internet and trying to resave and resave our figures. And for some reason, they're always just lower quality every time we try to resave them. And that can be incredibly frustrating. So the best format for saving your figure, in my opinion, is a TIFF. And this is because it has a characteristic called losslessness. And that means that the number and color of the pixels within your image are preserved through multiple saving iterations or alterations of the image. Again, this is very different from, say, a JPEG, which perhaps many of us are familiar with, where you click to save it as high resolution, you click save, and then the JPEG is very, very blurry when you look at it again, and that can be very frustrating. So again, you want to use vectors and you want to use TIFFs or EPS if possible. Another really big important component of kind of planning your figures starting from the beginning is making sure that you keep your original images and figure files. And again, what I mean is that if you're taking an image on a microscope, you want to make sure that that original high resolution, high quality image of whatever specimen or view that you take is saved by itself and unaltered throughout your entire process. Again, save it as an original, put it in a separate folder, duplicate that file, and then only start creating your revisions or adjustments from there so that you can always go back and reference that original file. And then just as a reference to note, most journals do require that images with text specifically, so if you have text overlaid on your images for labels, for example, be submitted at 600 DPI or higher. Sometimes images can be 300 DPI or higher, but again, you need to check with your journal guidelines. And there are also other requirements for different sort of line art submissions, which can be even higher resolution. So again, journal guidelines are a very important component of figure making. So again, this was step one, which is planning our figures. So kind of just thinking about what we want to create in our figures, what we're going to want to be showing, or what the expectations in our field are. Once we've done that, we're going to move on to step 2, which is prepping for creation. So this is before you're even starting to make your figures or only just starting to do so. First and foremost, read your target journal guidelines closely, specifically those for figure creation. Many journals can be sticklers on this, and unfortunately, it does vary journal to journal, which makes things very confusing for all of us. But again, once you select your target journal, read the guidelines related to the figures themselves. Again, this is where you're going to learn if there's a limitation on the number of figures that you can submit, how they should be numbered, if there are limitations or information related to the figure titles, to the legends, the image resolution, and what sort of file format the journal will accept the figures being submitted in. Another step here is to know your audience. And what I mean by that is you need to make sure that the figures you put forward in your manuscript not only correctly display the data, which is very, very important, but also conveys all the information that you want to the appropriate audience. And this is probably going to be quite different between a very specialized technical journal, where you probably have established scientists in the field that are reading it. They don't need as much background or context, versus, say, a high impact journal with an incredibly broad audience, where you might have to do maybe pictorial representations or schematics to really illustrate the big concepts that you're trying to put forward. So a good question to ask yourself when you're thinking about your target journal are your figures suitable for introductory readers or perhaps established scientists? And again, this is really going to depend a lot on the journal's aim and scope. One of the most important parts about prepping your figures for creation is identifying the role of each figure. So again, this is the purpose that each figure puts forward. So ask yourself, what is the underlying message of this figure in particular? How does it fit into the story of my manuscript? And from there, you can ask yourself a second question, which might be one that many of us struggle with, and that's how can I best express my data? What type of plot and graph am I going to be using? Again, this is something that journal editors and reviewers may comment on and would cause you to be recreating your figures and again, losing time down the road. There are several different standard visuals to choose from. The example here are just the little symbols that you might see in Microsoft Excel. And again, choosing the different options here to represent your data really depend on what you would like to communicate. So again, if you're looking to communicate a comparison, a distribution, trends over time, or a relationship, that's all going to be best done using a different sort of graph. And I have two examples here on the bottom right of the slide. So if you're saying comparing one set of values with another, you do have multiple options such as bar graphs, scatter plots, or data tables to do so. So you can start from there, but then perhaps ask your mentors or review the literature to see how other researchers have done it in your field and perhaps go from there. My other example here is the one that's also highlighted. So if you're looking to showcase a distribution of a set of values, again, you can use a scatter plot, a line graph, or a box and whisker plot. Again, this is going to be field specific, and there are always new and better ways to be showcasing your data. So again, just keeping up with the literature, discussing with your colleagues, and asking your mentors are a really good way to learn which types of plots and graphs are the best to express your data most clearly and concisely. Along similar lines is that before you create your figure, you need to identify which figure making tool or tools are going to be the best for you to use and also the most accessible for you. Importantly, there's often a dedicated or a commonly used tool that'll do exactly what you want to achieve in terms of your data representation. If you're a kind of an early career scientist or perhaps a graduate student only in your first couple of years, this might not be very obvious to you or clear, and that's totally fine. So if you are unsure, again, I encourage you to reach out to your colleagues, your advisors, or your mentors, or again, look to those method sections and the figures in papers that you're reading to kind of gain the background on your own research that you're doing. It's very, very important, and you will probably see it commonly, that people cite the software that they use to create their figures in their methods section. So again, just a little bit of reading and digging in the papers that you're referring to can point you in the right direction. So again, overall, the goal is to make few changes to those original images that you create. Again, those are your primary data, but you will have to make some adjustments, some polishing of those figures, and that's where these figure making tools come in. I have some examples here in case you're curious. The kind of two binary choices here are that there's going to be paid software that you can use to create your figures, and then there's going to be freeware. And these are only a very, very small amount of examples. Kind of the industry standard to create really beautiful figures are going to be associated with the Adobe Creative Suite, most importantly, Adobe Illustrator and Photoshop. However, these are expensive tools, but they may be something that you can get through your university. Other paid software options that are commonly used through a variety of fields to create figures include Microsoft Expression Design, CorelDRAW, MATLAB, and then Microsoft PowerPoint and Excel. However, there are free versions of Microsoft PowerPoint and Excel, so be aware of that. By contrast, free software options include GIMP, Dia, Inkscape, ImageJ, and R. So there are a lot of open source and freeware out there, and I think that's going to become more common in the industry moving forward. Again, so you may be able to find access to a lot of freeware, and there may be a lot of tutorials that are also free online for you to learn how to use these. And again, it might take some time investment on your part, but it will really pay off in the end. So now that we've planned our figures and we've prepped to create them, the last step is, of course, creating the figures for your publication or your talk or your poster. Again, I encourage you to revisit your target journal guidelines before you start creating your figures. Again, refresh yourself, become familiar with what you need to do. Again, following a journal-specific guidelines, especially in terms of the figures, can help you avoid that desk rejection. Journal editors are not going to be impressed if your figures are low quality. If you're trying to submit 12 figures instead of the allowed 8 figures, those things can lead to automatic rejection, which can be very, very frustrating. And however, it can also be very easily avoided before submission. Something that you should be aware of and double check within those journal guidelines are if there are any restrictions on figure width, figure type, and the resolution required by the journal. Does the journal allow or encourage color in figures? This may be something that is only important for journals that are still focusing a lot on print, which is not necessarily the case too much anymore. But if that is true, do they have, is the color space RGB or CYMK? That's going to be important. Font size and type that's allowed in figures, and then line weight if you have line graphs and things like that. So again, there are a lot of nitpicky little things that journals do pay attention to. So again, just double check to see if there's any information or guidance on these 5 points before starting to create your figures. My tips for when you're creating your figures are to present your data comprehensively. So remember that each piece of data has not only a description, but also a number, a unit, and probably an uncertainty. And as a result, you need to place all 4 components of those somehow in your figures. And if you decide that you're going to not show some certain data points, you need to be comprehensive and explain why. And if an error bar is present, you need to make sure that you explain what they represent. So again, you need to be very comprehensive for all data points shown in every figure. As I alluded to earlier, it's important to also be as objective as possible. A scientific figure should not mislead the reader. Yes, you can be declarative in terms of your primary interpretation, but you should not be adjusting your figure, revising it, or anything like that that could potentially be misleading. What we recommend is that you use the simplest type of plot that you can to convey your data accurately. And again, include relevant labels and values, but don't include so much information that it obscures your main message or your data itself. Along similar lines, it's important to avoid all unnecessary or confusing visual elements. So this could be too many colors, too many different labels, grid lines that don't need to be there, or too much color. And what I've highlighted here in orange is that many of the default outputs of graphs from many programs are probably not going to be satisfactory for your target journal. And what I've shown here is an example of a default output of a graph from Excel, and then one that should be submitted to the journal after some adjustments and modifications. Again, what you can see on the left compared to the right is that there's grid lines and dark coloring that was removed from the left towards the right. That little box that always appears on the right side of your Excel plots, that was removed. The text was made to be consistent and clear. And the data points were also put in front of the line because they're the most important elements of the plot. So again, this is an example of, again, how a default program may not be suitable. And other examples of this include outputs from MATLAB and R. Those are often ones that need a little bit of polishing before you submit. Another important point to consider when creating your figures is to use color effectively. So color shading, patterns, gradients, etc., can be really excellent to illustrate your main findings. However, the ones that, again, are automatically put out by the default often don't really look that good. They're not as visually appealing. And this is especially true when they're folded into a printed page in a manuscript within a journal. So again, I encourage you to use color to emphasize your message. But again, if you use green to represent series 1 in figure 1, you should be using green to represent series 1 throughout all your figures in your manuscript. So please be consistent across the different figures. It's important to avoid using low contrast colors, ones that are similar. If you use too similar of a color, two colors to do comparisons, it's not going to be clear for the reader. And it's also important to make sure that no information is lost if printed in grayscale. So again, color might not always be the best. You might need to use a dashed line versus a solid line, or a triangle icon versus a circular icon, to really make sure that your data can be clearly differentiated in print. So those were my main tips for creating effective figures. Again, there are many other points that Clark and I will be happy to discuss in the Q&A. So if you haven't put your questions in, I encourage you to do so now. What I want to kind of end here with is how we can write effective and clear figure legends, because figure legends are indeed not an afterthought. They are an integral part of your figure and instrumental for conveying your results. So the main objective of a figure legend is to make it possible for the reader to interpret and understand the significance of your figure without referring back to the main text. Again, to kind of bring home that point that journal editors, reviewers, and readers are going to jump straight for your figures and want to be able to understand the core message of your data and your research, basically right from those figures, and maybe with a little bit of the abstract and conclusions. So while formatting does differ from journal to journal, there are really 4 components that are essential to every figure legend. And I'm just going to review those briefly here. First, every figure legend needs a title. So say we have 4 panels in our figure. You don't want to jump into just explaining what each of those 4 panels means without including a title. So a title is a brief, succinct phrase that describes the entirety of the figure. So this is including all of the panels and all of the subfigures. You're showing all those panels in one figure because they're related, and the title should really indicate what that relationship is. Titles can generally be 2 types. The first is a declarative title, which summarizes the results or major findings portrayed in the figure. So the example I have here is that A mediates upregulation of B. So that's this declarative finding that you're putting forward in that title. The other option is a descriptive title, which may indicate the type of analysis used in the study. So for example, scanning electron microscope images of zircon-bearing geological samples. So you're giving a little bit more information in terms of what the reader is looking at and what methods were used in that analysis themselves. The second component of a figure legend probably has something to do with the methods that were associated in generating that data. So you should briefly describe the technique used or the experimental design. This does vary by journal. Some journals do not want methods in their legends. So again, refer to the guidelines for information. But if you are allowed to have some methodological information, this might include the animal model that you use, cell type, assay name, conditions, treatments analyzed, any statistics related to your work, or just the primary methods employed in general. Again, you only want to provide what's absolutely necessary for someone to understand the figure. So try not to be redundant with the main text, just what they would need to understand the figure without really going back to the main text for details. The third component that every figure legend should have is, of course, the primary result that the data is putting forward. You want to state the primary results that are shown in the figure. And again, this should be as concisely as possible, preferably 1 sentence or 2, or only a couple phrases. You can also include result-specific details. So if these are replicates, p-values, how many samples went into the averages, etc. This information can be included if applicable. Again, this can be in the legend or it can be up in the figure itself, but it should not be repeated between the two places. The last component a figure legend should include are definitions of abbreviations and acronyms. Again, this is going to be something that is journal specific, but if you're using any sort of abbreviation or acronym that's not standard to the field, this is something that needs to be explained to the readers themselves. So these elements, as well as any sort of not uncommon issues within your figures or elements of your figures, need to be explained. I do very strongly encourage, and if not, journals will require that you use the same abbreviations and nomenclature between the main text and the figures for consistency. So before we jump into the Q&A session, I do want to leave everybody with these 4 takeaways for creating eye-catching, visually appealing, accurate, and clear scientific figures for your manuscript. First, invest in your figures from the start of your research. And again, this is going to save you as authors so much time and a lot of frustration. You want to save your images in the right format, and you want to make sure that you keep all original files. Again, I recommend saving them as TIFF, so not as JPEGs or other low quality formats. This will kind of assist with saving you a lot of time and frustration associated with the first key takeaway. Before you even create your figures, you want to identify the purpose of each figure, the message it's conveying, and you want to use the right graph or plot or representation to put your data in place. This is something that I think comes with time and with experience. And again, do not feel like you can't reach out for information or guidance from your mentor, colleagues, or peers. And then last but definitely not least is that you should always be checking the journal guidelines before creating your figures. You do not want to spend time and effort creating a beautiful figure that you cannot actually end up submitting due to journal restrictions. So do yourself a favor, check the journal guidelines first, and then create your figure second. Beneath this, I do have three or four references that you guys can look at. And we do also have a lot of good articles on creating figures on the LetPub Learning Nexus, so check that out as well. And with that, I want to thank you for your attention and jump right into the Q&A with Clark. And just a reminder that we do have webinars each month, so I hope to see you soon for next month's webinar in June.
Thanks, Evie. Excellent as usual. We have plenty of activity both in chat and Q&A. I'm gonna I already have some questions lined up in Q&A that are great, but I'm sure more are going to get included in there now. So if you have questions, jump in the Q&A, leave them now. We already have a lot of upvotes, so it's like people have a lot of the same questions. So let's jump into it. And again, some of this you may have covered, as you went along with the presentation, because people asked some of these questions early, but we can just go over them again and maybe give a little bit more detail, answer some things a little bit more specifically. So first one here that's quite popular regarding colors. So if I want to plot a figure with many different colors, could you please recommend some color combinations or guidelines for color combinations for a single figure? There's obviously a concern here for aesthetics and clarity and the ability to, of course, distinguish between those different colors. So any more additional comments on how you're choosing colors?
I think that's a really good question, and I think it is journal dependent and author dependent and data dependent as well. Again, you want to avoid using colors that are from a very similar tone and hue and family. So I would say, you don't want to show 3 different data types with red, pink, and peach or something like that. You want to make sure that the colors are drastically different from one another. So use like red, a darker color, blue or black. So think about them as being very high contrast. So black versus white. That's going to be a very clear binary comparison that you can put forward. I don't have any specific great combinations that I would always use. You don't want to use so many different colors that it can be distracting to the reader. They need to be complementary. But that I suggest actually just doing a little bit of research on the internet. There are a lot of blogs out there that you can reference. For that. I mean, Clark, do you have any good color combos that you used to always just jump into to show your data?
Yeah, one thing I want to, because I've written articles before on selecting colors mostly for accessibility issues, right? Because
Right,
there's
yeah.
people that are colorblind. And so, but I find those to just be better following those guidelines. So I'm going to put one here in the chat so you guys can see that one from ASCB. They're just good in general for picking good colors because science had a problem at one time where it was constantly using green and red, right? This is like very like popular for imaging studies. They'll use like green and red. And one, it's not good for accessibility for colorblind scientists. And two, it's just ugly, in my opinion, if you're going to choose a color. Those two don't really work as well as people think. Aesthetics and clarity looking between red and green. So if you look at that article, they make comments about using like cyan, magenta, these types of things, but there's plenty of combinations in there. Also, what I would say is using color to make it look good, I think is not a route you want to go. So you should be asking yourself whether you want to use color or not, and you need a good reason to use it. And you'll see that the journals encourage this, right? So colored figures are usually gonna be an additional expense and everything. So look into that first and decide whether you want it. In science, clarity is beauty. So focus on whether you need to be using color. Grayscale is quite often used, and you don't have any accessibility issues. And I think it's quite clear when you use grayscale. As you mentioned, AV, if they do use patterning, like cross-hatching and things like this, the defaults that your program, your software may use may not be good, so you'll have to tweak it. But consider whether color is really necessary, I think, is the first key. And aim for clarity first and foremost. But other than that, you can look at the color spectrums given in there. So heat maps are a common thing, I think. And again, the default colors used by the heat map, particularly if it's like a red and green, I don't think is good. So go ahead and check out that link, and I can post a few others as we go through these questions. I'll also leave them on the PureRef discussion board, which I posted in the chat a couple of times. We can leave some more links there. Okay, so that one's all set. Is there a standard font size to be used upon creating a figure?
Again, everything differs journal to journal, but I think some of the most straightforward fonts that you've probably used before, like Arial, for example, size 10-point font at minimum is preferred. So again, you should be able to print out your figures at 100%, which is something that is always a good tip to do. Make your figures, print them out at 100%, and look at it like you'd be reading a paper on your desk. And if you can't read the font, then it's too small. If you're straining your eyes a little bit, it's too small. So do those like quick little steps, and I think that'll help you really identify what fonts are best. I think Helvetica is probably another good font. Clark, do you have any favorites?
My favorite, I guess it's more based on approach, right? And so exactly what you said right there is what we recommend during our editing projects to authors all the time is the eye test, right? That's the whole point of it, is to be readable. So I think for me, it's more approach based. So I look at it, I'm submitting to this journal. If the journal tells you to have the figures in separate files, right? And the legends on separate, right? So it's all uniform, then it's just easy. You just do whatever Times New Roman, 12-point, whatever they specify as the font for the manuscript text. I usually just use that, right, for like the legends and the text and the figures, because they're all independent. If it's one where they want the figures embedded and it's a little bit closer what it's going to be in production, then I do, as you said, I choose a font that makes sense when I'm viewing the paper. So whatever I would like to view the paper at to make the body text legible, I then choose a corresponding font for the figures, because all that matters is the experience of the reviewers, right, and the experience of the editor. So make sure that experience is good, because they're gonna, none of this matters, because in production, once it's accepted, they're gonna choose it anyway. So you're just trying to align with either what they specify or with what's the most readable, the clearest for the reviewers. So check that first. If they ask you to separate it out, if they give clear indications of font from the manuscript, go with that. If they leave it a bit ambiguous, go ahead and just do the eye test and pick something that's the most readable. But yeah, that's a big thing, because we see it all the time. Authors will put stuff that's just clearly illegible, right? And that doesn't work. If you can't read it when you open the file, do not send that to reviewer. So yeah, eye test is big here.
Yeah, that's a very big frustration for journal editors and reviewers. And again, I think that, that goes into the point that I was trying to make, and that you made too, making your figures as clear as possible is important. And that means not trying to include tons and tons of text on your figures. Again, save that for the figure legends, try to pare it down if you can, but only the most critical and relevant labels need to be included on your figures in text.
Okay, next question. What's your opinion on PowerPoint as a tool for figure formatting?
I think that it's a fine tool if that is what you have access to. I've definitely made posters for conferences in PowerPoint before I had access to Adobe, and I think that sometimes we just have to work with what we have. So it is something that you can use. I do, however, think that there are a lot of newly evolving or even just already out there freeware programs that you can gain access to that will make more polished figures than PowerPoint. PowerPoint is limited in many aspects for figure making. So if you can gain access to other programs, I would suggest doing so, but I wouldn't write PowerPoint off if that's all that you have.
Yeah, I agree completely. So I'm not a big fan of PowerPoint and Excel just because journals often say professional graphing software, and generally that software is not designed for graphing. It's a feature that you do after, and it's commonly seen, it's commonly utilized. Like my biomechanics students, my comparative physiology students, we were in lab, we would have them make things in Excel, right? So those are, like you said, very, very usable, but because we're scientists, we're all about optimization. And it's these little red flags that a reviewer or the editor sees it and sees that you've elected to use that rather than a better option. It's just so competitive that when they look at your submission, they're looking at everything and the decisions you made, this holistic view of, is this person optimizing this study and this manuscript? And it's one of those things, when it's unoptimized, it just leaves a little bit of a bad impression. And so if possible, I would do something else. I don't think it's a major problem. You can do it with PowerPoint and everything, like you said, particularly if that's what you have available. But we hope that you come to a presentation like this, AB presents all these other great options to you. See if you can adopt some of those other options and give you a leg up. That's what you're here for, right? Is to improve your figure making. So I would suggest, if you're using PowerPoint, look at some of these other options because it may give you a little advantage moving forward.
Yeah, check out how figures are made in the references or in the manuscripts that you're referencing and go from there. That will probably give you some good clues about the standard programs used in your field.
Yep. So workable, but we're always trying to optimize. Very good question here. So usually we have to try several different journals before getting accepted. And this user says we, like their lab, but I think we speaks for the community here. So does it waste too much time to customize the format to accommodate that specific journals? I've heard that editors may not reject a paper purely by its format. So thoughts on this topic, which is pretty expansive.
Yeah, it's an expansive one for sure. I think a lot of what I discussed in guidance and in webinar and everything today is something that can be generally applied for submissions to all journals. It's really just those specific ones that are super nitpicky about everything that can, be roadblocks to you or time-consuming issues. Again, if you have, like an example is paneling. Paneling is going to save you the number of figures, and paneling is going to be something that's accepted by all journals. However, maybe one journal wants capital letters, A, B, C, and D, and the other journal wants lowercase letters. Those are going to be small, easy fixes for you to do. If you really want one figure that you spent so much time on to submit to one journal, and then that figure is the one that exceeds the limits for the next journal that you're submitting to, don't be afraid to ask the journal editor and explain why that figure is so essential to your manuscript and should be included. They will make exceptions as long as you provide justification. Again, there's also the opportunity to use the supplementary material for submitting figures. And oftentimes those figures that are in the supplementary don't have to be formatted the exact same way as the main text. So if you really don't want to change a figure, you can't change a figure for some reason, you may be able to actually get it published by putting it in the supplementary, because that won't require the formatting. So that's like a little trick that can happen. Not saying that that's something that you can get by for all journals. You always have to check, but that could save you a little bit of time.
Yeah, I have a couple of points. I'll try to be brief on this one, but I agree completely. I do like that a little bit of a trick that you indicated there. For this, what I think is, so certainly if the journal has specific formatting requirements, you simply have to follow those. I find that most of those formatting requirements are specific to how they're submitted in terms of separated out, this type of thing, and like font size and stuff like that. So that's relatively easy if you're modifying that formatting. Otherwise, they don't usually have a lot of formatting restrictions, and you can get by with just making strong figures. So if you're prepping your figures, just make strong figures that are going to be readable, right, and clear for the reviewers, and then you can usually get by with it after the submission phase, whatever changes need to be made. With that in mind, I think this is really more of a question of journal selection, right, than it is of formatting. So this is just based on the journal, and as such, if this is a concern for you, you should be carefully thinking about where you want to submit. Lots and lots of journals are going to a model where they have less formatting restrictions for submission because this is a problem. Maybe you want to lean more towards those journals. When you're prepping, generally you should have an idea of the journals you want to submit to in mind at first. Format to the strictest journal first, or the one that you, if the one that you're hoping to get into, maybe your top 1 or 2 choices, whichever one has the strict formatting, format to that, and then you can format away from it right afterwards. So, and these are the things to think about. It really comes down to journal selection, and your whole holistic view on submission process is what's going to be important here, because yeah, you can't get around it sometimes, but I would prefer to just make strong figures and then figure it out from there, and maybe change my submission practices based on that.
Yeah, that's good advice for sure.
Quick, easy one for you, specific to geology. What's your recommended software for your field for figure creation?
For geology, you're probably going to run into ArcGIS, which is very, very common in a lot of geosciences. ArcGIS, however, is not free, but there's now a new program called QGIS, which is free and compatible with both Mac and PC platforms, which is very, very good, because Arc was originally only compatible with PC. This is a software that I think everybody as a geologist needs to know if you're in consulting or you go into industry as well. However, it is definitely an example of the default images that Arc puts out is not what you want to submit for your final figure. You will have to make adjustments. So it's very similar to the Excel situation that Clark just went through. Other common platforms would be R and MATLAB. Those will probably be good figure creation software. And then what I used to always use would be the Adobe suite. So I made all of my figures, polished them up in Adobe Illustrator. And then if I had any sort of field photos and images that I wanted to put in, I would make sure that they were made as polished as possible in Photoshop before creating the paneled figure itself in Illustrator. So those is what I would say for geology. Yeah, other kind of data analysis software for like remote sensing and things like that, Envy would be good. But again, that's something that you'd have to use a secondary software for polishing. So I'll leave it at that, but if you have more questions related to geology, of course, I'm happy to answer. Just throw those into the PeerRef chat and I can provide some links for access.
Yeah, if you have specific things like that, definitely jump over the hub with us throughout this next month. You can get real specific answers from myself or AB or other users. Is it allowable to use axis breaks in figures?
That's a good question. That might be journal dependent. I don't know what. I guess I never have had to do so in mine before. Have you, Clark?
Yeah. So axis breaks or like truncating axes. I think this is a big question. There's often an example used here where it demonstrates that when you significantly truncate axes, you can make something look like a difference, right? And the tricky part here is you're going to be judged based on perspective of what you're trying to do. And so always elect to make these changes the way in terms of the way you represent the data consistent with what the data is actually saying. if you try to make change, like if you truncate the axes and you have non-significant differences, or sorry, no difference, then you're doing something, you're representing the data in a way that it's not actually speaking to. And so you can always truncate axes when it's necessary to show the visual difference that matches the statistical difference, right? Truncating axes is definitely allowed, I think. I don't think it's like banned or like any journals like including that as like something that's like a formatting thing that you cannot do. But if you do it in an inappropriate way, certainly the reviewers are going to be very, very critical of you. It's the same thing with things like including standard error or measures of dispersion, right, on your figures. There's no rule, but if you fail to include it, I think there's a good chance that a reviewer is going to be perturbed by that. So yeah, there's no rule against it, but you should be using it appropriately, I think is the key there. And I've used truncated axes all the time for that very reason. It's simply the scale is like way too large, and to show the shape, particularly of like line graphs and things like this, you want to truncate the axes, but the extent to which you do it and the way you do it to show whether there are differences or not differences, that's the key, how it's utilized. Okay. So there's two related questions, and so I'm going to do them both here. The questions basically come down to is, can I use sort of visually compelling figures, but the key term here is used as fancy, eye-catching, beautiful, in order to increase my chances of acceptance, or should it just be compatible with the content or simple or ordinary? Both questions are essentially asking the same thing. We want to make, and this goes along with the color question we had earlier, people want to make there's this concern that figures are an opportunity for aesthetic appeal. What's your opinion on this, AV?
They need to be compatible. They need to demonstrate your data clearly and effectively and accurately. That is more important than I think making them beautiful and visually appealing. I think that the visual appeal and making them aesthetically pleasing is something that is appreciated. And as you just said, Clark, figures are an opportunity to kind of introduce that into the scientific component of the manuscript itself. Again, that might be something at first glance that brings a reader into your manuscript or gets you past that first view from the journal editor, and kind of, they appreciate that you optimized your figures and that you put the time to do that. But if they see, they'll be able to see right through if your figures are just colorful and artfully made, but the science isn't sound. The reviewers and editors will be able to see right through that. So my opinion is, first and foremost, make the figures clear, ordinary, and just represent your data the best way possible. And that might be a little boring, but I think that is way more important than making them fancy, so to speak.
Yeah, the figures can be very powerful, but they're powerful when you glance at the figure and you get the whole idea of the conclusion right there. That's what makes a figure powerful. I don't think like aesthetic appeal, like the color or a fancy style, like a volcano plot or like anything like this, trying to insert a fancy style, or a lot of these like 3-axis graphs and things like that, they can make sense at times. But if you're trying to force things like that, I think people get themselves into a ton of trouble with this. I think they, as a reviewer, I see right through it all the time. As an editor, like if you do I expect a certain graph. I read your data and then I expect a certain graph. And what really wows me is when I see the graph and I go, oh, like perfect, like a great visual representation. Like this summarizes what I was thinking when I was looking at your data. But if I see you throw in a figure and it's dressed up in this way, and I can clearly see that you're focused on like making the figure like look bright and showy or something like that, it instantly like lowers credibility with me, and it actually turns me off to your data. I think science is very standardized, and when I'm reading your manuscript, I expect certain figures, and if I see something outside that and you're trying to do something with it, something fancy, And it actually, it's a huge blow to you, I think, as an author and a submission. So I think people get themselves in trouble here. And in my opinion, I would really avoid that. Like, figure out what people expect to see and then meet their expectations in a very meticulous and careful way. That's what I think reviewers and editors want to see. Again, beauty is in clarity in science. And so do the figure they expect to see and do it really well, execute it well, but don't do anything fancy. The one thing I will say though is sometimes you do have an opportunity for a sort of summary figure, a schematic, something like this. Those can give you a little bit of opportunity to, but again, it's doing the same thing. It's just showing the reader like, oh, based on the method I was looking at, this is a great visual representation of it. So it's doing the same thing as what I indicated before. But those types of figures, they are maybe an opportunity where you can make it look beautiful, where there's a little bit of illustrative nature to it and it can give you a little advantage. Yeah, that's a great opportunity. But otherwise, if like you can't use figures for aesthetics, do journal cover art if you're looking for aesthetics, because it just doesn't work. It red flags it for a lot of reviewers.
Yeah, I think people get bogged down too by trying to make things beautiful, and they add all those colors and lines and different elements that aren't necessarily relevant or even appropriate. And again, those are those elements that I was referring to about making you're transforming this clear figure that you created, and then you're making it confusing by adding all of these artsy elements to it. So again, add what you need and what the reader needs to understand the figure as quickly and as effectively as possible. And I think that'll be something that doesn't frustrate any readers or reviewers.
Yeah, you're not going for wow effect. The wow effect is moment of clarity, giving that person a moment of clarity. And you give that clarity by aligning with what they were reading in the manuscript. And if you over format the figures, right, then it doesn't align with the manuscript. It's jarring. And yeah, it's not the way to go. I think people get themselves in trouble thinking about figures as beautiful. There's a lot of opportunities for data visualization to be beautiful, but mostly in scientific manuscripts for reviewers and editors, it's an intuitiveness that really appeals to them. So aim for that.
What do you think we have time for? Maybe one more question?
Yes. Well, one basic one here is there a limit to the length of legends, journal specific. That's always going to be in your author guidelines. Just go to your author guidelines. Send a quick email to the journal if you're unsure of it. But that's just like abstracts. That's just very specific to the journal.
Yeah, that could be quite common for high impact journals, nature, science, things like that. A lot of people, because there's such a strict limit on the word count of the text, way back, years ago and things like that, people tried to pack in so much information into the figure legends. And as a result, they're very strict with their guidelines now. So just be aware of that.
Can you give me opinions on two other pieces of software, SPSS and Stata?
I've actually not used either in my field. So I know that SPSS is something that you probably use, Clark.
So I've used both. They're fine. They're good. SPSS and Stata both work fine. In particular, this is also one thing that may be helpful for people that have this, because these could be endless, right? People can ask us, like, what's your opinion on this software, this software? One good rule of thumb that I think it can be quite helpful is if the piece of software is used for statistical analysis, it's probably pretty good as the graphing software. You might have different preferences and it might have features that you want, but in terms of saying, like, is this legitimate? Right? Is it going to be acceptable? I think that's a good sign, right? So like typically that's why I don't really like Excel either, because typically you're not doing like your statistical analysis in Excel. So I wouldn't use that as like a piece of graphing software. SPSS, Stata, they both run the analysis. And so using them also as you're plotting, I think is completely fine. So I don't have any others that are super pertinent right now that I think we need to address. The rest we can get to you guys in the discussion hub. So we will be sure, let me paste it one more time for you. Go ahead and visit us in this discussion hub that I'm putting in chat right now. So go ahead there, ask us questions, have a back and forth discussion with us. We have like, I think 3,000 users or something in that hub. So there's plenty of people in there. If you want to crowdsource answers to these questions, that's definitely the place to go. Couple of housekeeping reminders before we go as well. Give us 24 hours and you're likely to get a follow-up email that's going to have this entire recording. So if there's any, I know a lot of people have information from AV's presentation that they want to know, particularly like the software recommendations, that type of thing, any of her references. You will get this on-demand recording. Feel free to share that with colleagues as well who might have missed this. You'll also get the certificate of completion for this webinar. So you get a PDF. You'll just have to take it as is, but it's a PDF that's going to have the name and date of this presentation if that's something that you can use for sort of continuing education credit. So I just wanted to remind you of those two things. AV, as you mentioned, we will have a webinar again next month like we do every month. So please check your inboxes to get the email for that. We don't have our topic set up yet, but we'll cook up something good for you and have another webinar for you. Any final thoughts or
no, again, just if you continue to have questions about this webinar, again, you can ask those questions all throughout the month on PRF. So just jump in the hub, ask your question. As Clark said, crowdsource from others. You know, Clark and I may not be in your immediate field, but it's very likely that someone that is within the hub with us is in your immediate field, and you can ask them directly and have a conversation about how they make figures, and you can learn from each other. So explore PRF, and we hope to see you guys all next month.
Yes. Thank you so much for the content, AV. We really appreciate it. Thank you to our viewers for coming on. Over 600 people today. It was a great crowd. We really appreciate it, and we hope to see you next month.
All right.
Take
See you
care.
next month.